Good morning, everyone. Today, my topic is non-equilibrium thermodynamics in biophysics. Non-equilibrium thermodynamics play a critical role in the biological system, as the living organisms are continuously exchanging energy and matter with their surrounding. Unlike systems in the equilibrium, biological processes are characterized by gradients like concentration, temperature, pressure, and continuously moving toward the maintaining non-equilibrium state of such life. Here we explore the functional concept of the information and the entropy in the non-equilibrium thermodynamics in the biology and examine some of the equations that describe non-equilibrium process. So now let's discuss our first topic, information and entropy. Entropy is a measuring of the disorder in the system. In thermodynamics, entropy is referred to the number of possibility configuration on the system, which highly entropy, including dis- disorder or randomness. The second law of thermodynamics states the isolation system entropy enter to increase lead to the equilibrium state. In biological system, entropy role is understanding how cell maintain energy and maintain order by improving energy. For example, nutrient or sunlight system decreasing entropy local example by organized molecule but increasing entropy globally by releasing energy. Some key equation in entropy and information theory. The relationship between entropy and information is critical for biological systems, especially in the contrast of genetic information and cellular organization. Boltzmann entropy is equal to Kz in delta, where the H is the entropy, Kb is the Boltzmann constant, and it represents the number of possibility microstate of the system. Biological systems maintain microstate carefully by maintaining low entropy locally, distributing increasing entropy over in the environment. Second equation is Sampson entropy, where H is Sampson entropy, Pi is the probability of each state I. Here you can see the formula. In here, Sampson entropy is essential in the biological system, especially in the genetic and the information process, in equating the amount of information and helping in understanding how organisms encode and process information. The third one is GABA free energy and entropy. Biological systems rely on GABA free energy G to determine spontaneity in the process. Delta GABA equal to delta H minus D delta H, where delta G is the change in the GABA free energy, delta H is the change in the enthalpy, and T is the temperature, and delta S is the change in the entropy. Negative delta G, including a spontaneous process vital for cellular process that require energy coupling. So our next topic is non-equilibrium process. Non-equilibrium thermodynamics describe how systems move toward the equilibrium and how they maintain non-equilibrium state in biology. Non-equilibrium process allow for metabolism, signal transduction, and other life sustaining function. A critical function of non-equilibrium system is irreversibility, as the biological reactions are often derived by chemical gradient and energy destination. So their key concept and equation in non-equilibrium process are flux and force relationship. Non-equilibrium system often feature flux. Example, flow of ion across the membrane, and the thermodynamic force concentration gradient that derive these fluid, where J naught is the flux example of the ion or the molecule. Xj is the thermodynamic force deriving Ji, and Lij are the coefficient that depending on the system process. Biological system depending on these relationships such as protein gradient in mitochondria derive atom ATP synthesis. Another one is entropy production rate. The rate of the entropy production equivalent the utility in the non-equilibrium process, where J naught represents flux, Xi represents force. This is the formula. So positive entropy process is a change characteristic of the non-equilibrium process and that distribute how the biological system distributes energy to maintain organic system. Onsager reciprocal relationship. In this, the near equilibrium continue onsetting polar relationship deriving the linear relationship between flux and force. The system show the cross-coupling process example couple transmission of the molecule can occur efficiently improving for action transfer in the cell. Chemical potential in non-equilibrium system. The chemical potential mu derive chemical relationship and diffusion. Here is the formula where UI is chemical potential, Ci is the concentration of the species I, R is the gas concentrate, T in the temperature. Difference in the chemical potential example across the cell membrane derive the difference of the molecule and osmotic pressure, especially for the nutrient intake and waste removal. Now let's discuss some biological example of non-equilibrium thermodynamic ATP synthesis. The proton gradient across mitochondria membrane is the non-equilibrium state maintained by the electron transfer chain. ATP synthesis use this gradient to convert ADP to ATP, deriving by the flux-4 relationship. Ion transport in neurons. Action potential depending on the concentration gradient of the sodium and potassium ion across cell membrane. Non-equilibrium conditions show the rapid significant by maintaining this gradient. And the last one is photosynthesis. In photosynthetic organism, light energy create a electron gradient, especially in a non-equilibrium state. And this granule derive the concentration of CO2 into the organic molecule, releasing oxygen as a byproduct. Thank you for listening me.